Great, and so I'd like to present our final main speaker of this session. So Professor Hugh Markus is the Professor of Stroke and Medicine and Honorary Consultant Neurologist in the Department of Clinical Neurosciences here at the University of Cambridge. And today he's going to talk about vascular contributions to dementia, the neglected player. I shall turn that on.
Thank you very much. Unfortunately, I've got a temporary problem walking, so I have to give this talk sitting down today. So we've been talking about the burden of dementia, and the figures are really worrying, particularly as you can see in low and middle income countries. But if you look at the data in a little bit more detail, it's interesting that the chance that one of us will get dementia is actually going down. So although the numbers are going up because we've got so many elderly people, the incidence is actually going down. And it's interesting looking at this data, from the point of view of a cardiovascular physician, that these prevalence figures, which have been shown in Europe and in the US and other places, mirror very much what we've seen in cardiovascular disease. So we've seen a really dramatic reduction in mortality and incidence of cardiovascular disease. If you look at heart attack, that's about a halving, and stroke has come progressively down. We don't know completely why this is, whether it's treatments or whether it's actually better lifestyle and so on, but it's a pretty dramatic change, a great success. So this sort of raises the possibility as to whether part of this reduction that we're seeing in the incidence of all types of dementia could be cardiovascular related. And indeed, there's a lot of data to suggest that risk factors, cardiovascular risk factors, are risk factors for all dementias, including Alzheimer's. But of course, it's difficult to tease this out, and causality in these relationships is very difficult. But there has been some really important data that came out a few years ago, I think, from the SPRINT trial. And this was a cardiovascular trial in which the investigators were looking at whether if you hit blood pressure really hard, going down to levels of 120 as opposed to 140, you could reduce cardiovascular events. And indeed you could by 25%. But what they did, which was a really, really good thing, I hope many more studies will do, is they included a cognitive arm. So they took all these people and did cognitive testing, and also in a subgroup did MRI and other imaging. And of course, it took a bit longer to get this result, but you can see that over time, it looks like intensive blood pressure lowering is associated with a reduction in cognitive impairment. And the cumulative endpoint was a mild cognitive impairment and dementia together. I've blown them up here. So you can see there's about a 20% reduction in dementia. This is a big effect. When you compare this with some of the treatments we've been talking about today, it matches those. The dementia isn't quite significant, probably because the follow-up isn't quite long enough, because you need a really long follow-up. So I think this has transformed the way we look at it, because we now know that treating vascular risk can reduce all types of dementia. This is a second study that came out about 2-3 weeks ago, which looks at population-based cohorts and suggests that treating dementia, even in people in their 60s, 70s, and 80s, actually treating hypertension actually reduces the risk of subsequent dementia. So really quite encouraging that we can do something about dementia by treating vascular risk. So how could this work? So there are two real possibilities. We've heard about cognitive reserve from Barbara. One possibility is that if you've got multiple processes damaging the brain, one of which is vascular damage and strokes and so on, this will reduce your cognitive reserve, so you can cope less well with the neurodegenerative pathology you're going to get. The second is there could be some direct interaction between these vascular factors and Alzheimer's pathology. And there's been a lot of interest in this area, and I'm not going to talk about it today. This is an excellent review on the topic. But there have been suggestions that perhaps the vascular risk factors may impair drainage of toxins like amyloid and tau, and so on. So there may be direct effects. But what I'm going to talk about today is a particular interest of our group, which is another mechanism by which I think this effect is mediated, which is small vessel disease. So this is a disease of the small perforating vessels, which you can see in the brain here. They come off the large vessels and they penetrate down into the white matter and deep grey matter nuclei. And the two cardinal features are acute strokes, here you can see one in the thalamus, and more diffuse white matter changes, which we call white matter hyperintensities on T2 MRI. Now, excitingly, you can actually see this process in man now on 70 MRI, which we're lucky enough to have at the WIBIC. So here you can see a healthy volunteer. Here's a circle of Willis. Here's your middle cerebral artery. Here you can see normal perforating arteries coming off, looking pretty healthy, coming off at roughly right angles. Here you can see a patient with hypertensive small vessel disease with, for example, a very tortuous small perforating artery here. And this disease causes damage in a number of ways to the brain. We've already talked about the acute infarcts. We've talked about the white matter disease. You get much more diffuse white matter disease affecting the whole white matter. In fact, if you use a technique called diffusion tensor imaging to look at white matter ultrastructure, you get enlarged perivascular spaces. There's a lot of interest in how these contribute to brain drainage. You get small microbleeds, and you get brain atrophy. And it's a very prevalent problem. So if you look at white matter hyperintensities on community populations, about 10 to 20 people in their 60s will have these changes. By the time you get up to 80s or 90s, it's almost present in everybody. So you could almost think of it as a sort of age-related change, which is more exaggerated in some people. So cerebral small vessel disease causes a cause for all strokes. But the reason we're talking about it today is because it's the most common cause of vascular dementia. Here are a few politicians who have suffered from small vessel disease, all three of these, and probably Margaret Thatcher as well, so common problem. But that's not all. It doesn't just cause vascular dementia. It contributes to the chance that if you've got a neurodegenerative pathology, you're going to get dementia. So if this is actually, although this is really only coming into the fore now, it's been known about for a long time. So this is a very beautiful study called the Nun study in the US, in which order of nuns were followed during life, had cognitive testing, gave their brains after death. And this slide shows the proportion of nuns who had Alzheimer's pathology at death, who had dementia during life. And you can see that if you've got Alzheimer's pathology, you've only got about a 50% chance of having dementia in life. But if you've got small vessel disease, it goes up to 90%. So there's this really strong interaction between the two. And this is really important because most people who have dementia, most of the people that Ben would see, for example, will be elderly people. And lots of data from multiple studies, CFAS in the UK and also this study from the US, shows that the vast majority of these people have got multiple pathologies. So they don't just have AD pathology, they've got 2, 3, 4 pathologies, all contributing to the dementia. And one of the very commonest of these is small vessel disease. So you can see how this small vessel disease is contributing to a lot of the dementia burden, including the Alzheimer's and other dementia burdens we're seeing today. So you might ask why you don't know more about it. Why hasn't it been focused on more? Well, I think we have had a problem in the UK that most dementia research has been focused on the very traditional dementia pathways. And this is reflected in the lack of funding. You only need to look at how much the DRI has spent on the vascular component, or the DPUK, compared with work on amyloid, and it's a very, very marked difference. But there are other problems as well. One is trial methodology. So because it's not such an easy concept, we need good endpoints for drug companies, etc. And this has been one thing that's limited drug company engagement in the area. And very importantly, a lack of therapeutic targets. So we've been working to try and improve trial methodology. This was an international consensus that we had last year to try and develop a pathway to take this disease or trials in the disease all the way through from more phase 2 trials using surrogate endpoints. MRI is a very good surrogate endpoint for this disease, particularly DTI, right through to clinical endpoints. So hopefully this will encourage companies and get more involved in this area. But the area I'd like to just finish talking a little bit about is therapeutic targets. And it's a very difficult disease to study because most of these strokes are quite minor. People don't usually die of them acutely. So by the time we get the brains, that end stage brains, we're not getting tissue from early on in the disease. So it's quite difficult to know what's going on in the initiating events of the disease. But this field has been really transformed in the last sort of 4 or 5 years by genetics. And a variety of techniques are used, but mostly the genome-wide association study, which most of you will be familiar with, but you genotype up to a million or more polymorphisms spanning the whole of the chromosome. Then you compare the association of each of these polymorphisms between cases and controls, and then you can identify regions of the chromosome that are associated with disease. And this disease has been a little bit late coming to the table, but we now have a lot of data. So this is data that Matthew Traylor published a couple of years ago, looking at lacunar strokes, showing 12 loci. We've got a lot of loci for white matter hyperintensity and DT-anti-Saladin Persons work, looking at this. And recently we've had a very interesting paper looking at perivascular spaces from the group in Stephanie Bette's group in Bordeaux, looking at finding 12 more loci. So we've got a huge amount of information coming out of this. But one area that seems to be particularly promising is the neurovascular unit. So quite a few of the genetic associations we're getting are focusing on the neurovascular unit. So what is this? So the neurovascular unit is really the sort of complex of these very tiny vessels in the brain. And it's composed of your endothelial cells, your brain endothelial cells. You've then got your basement membrane here, and then you've got your mural cells or pericytes. And then surrounding these are the astrocytes. And a number of abnormalities could occur in small vessel disease to disrupt this process, and particularly extracellular matrix breakdown. So you can imagine if your matrix breaks down, you're going to get leaky small vessels, you're going to get leakage of tissue from the serum into the brain, fluids from the serum into the brain. And this is just some of the data from one of the papers showing that many of the genes that we're getting seem to play a key role in extracellular matrix homeostasis. So this is a project we collaborated with Karen Horsburgh from Edinburgh, who developed a beautiful technique to pull out the extracellular matrix from human brains by enriching it, and then carrying out proteomic approaches, and then being able to look at all the identify all the underlying proteins and genes involved in the human extracellular matrix, and then superimposing this data with the data we're getting out of our genetic data, and showing that there is a really strong overlap between the genetic data we're getting out and the networks of extracellular matrix proteins and associated proteins. So again, provide more evidence implicating extracellular matrix protein dysfunction in small vessel disease. And we've also got data from man to suggest that these processes are important. So it's now possible to measure blood-brain barrier permeability in man using a MR-based technique. So you give a dose of gadolinium. You then follow the patient up with imaging for about 30 minutes, because this is very slow, low-grade leakage, so that you need to monitor for quite a time. And then you can look at movement of the signal, gadolinium-based signal, into the brain, and you can show that you get increased leakage in patients with small vessel disease. And particularly recently, using a technique developed by Gary Rosenberg in the States, it's actually possible to get spatial maps of blood-brain barrier leakage. So here, the green is the white matter hyperintensity. Here, the red is areas of blood-brain barrier leakage, which you can see is happening sort of on the edges of these white matter hyperintensities. So it looks like this is important in man too. So we've been interested in trying to take this through to treatment, and this is work that Alex Granata, who leads runs the iPSC lab in our group, is taking forward. So in using iPSC technology, you can take cells from the patients who I see in my clinic with many genetic forms of small vessel disease, and then you can reprogram these into, in this case, brain endothelial cells from the neurovascular unit and also neural cells. And then you can put these into a model and look at the relationships between the two. So this is just an example, for example, where we've taken patients who have got COL4A mutations. So this is one of the genes that can both cause monogenic and is associated with polygenic small vessel disease. And you can show that if you look at the cells in this model, you can show that they've got pathological changes consistent with extracellular matrix disruption. And you could also look at the permeability of the endothelial layer and show that the permeability is disrupted in these cells, but not in corrected control cells. So it does look as if we're mimicking what is going on in the human in this situation. And this is something that I find very interesting, some of Alex's data, which shows that if you take the mural cells and you take the media off the mural cells but leave the mural cells behind, you can transfer this pathological effect into the endothelial cells. So there's some sort of paracrine effect that's being transmitted in the supernatant into these endothelial cells, which leads them to then develop pathological changes and also increase permeability. So this is really exciting. It suggests that if we could identify what this is, perhaps we can inhibit it and prevent disease progression. So what we're now doing is trying to move this on to look at drug screening, and for various reasons, we've decided to focus initially on matrix metalloproteinases. This is on the basis of genetic data, the data from the Karen Horsburgh's study, and also data from Alex's study, looking at expression of different proteins, both in the supernatant and in the tissue in her model. And initial studies, taking compounds from AstraZeneca Open Target Library, it's been possible to reverse both these endothelial changes and these permeability changes using inhibitors of matrix metalloproteinases. And this is very, very early data, and we need to look at this in a lot more detail and then translate it into other models. But it's certainly very exciting and suggests that perhaps we can inhibit the processes of causing small vessel disease for the first time. So I'd like to stop there, but just, I think vascular risk in dementia has been very under-recognized. It's now beginning to be recognized, but sadly not as much in the UK as in other areas. We really need more work on it. Recent data suggests treating vascular risk factors reduces dementia. The evidence for hypertension now, I think, is overwhelming. We need more data for other risk factors. Much of this association, I think, is accounted for by small vessel disease. It's a really a ripe area for research investment. And we've got some really exciting advances coming through, and so many different potential pathways that could be followed up, which are suggesting novel treatment approaches. So just lastly, I'd like to thank the whole of the research team and Alex, who couldn't be here on this day, and one imposter you'll recognize, and all our local, national, and international collaborators and the funders. And just to remind you to go home and measure your blood pressure. Thank you very much.